I'm here today with Leslie Thompson, one of the world's leading authorities in Huntington's disease. Leslie, I wonder if you might describe for people what Huntington's disease is.
Huntington's disease, or HD, is an inherited neurodegenerative disease, so it causes brain cells to become sick and die. It's a truly devastating disease that strikes people typically in the prime of their life. And it has sort of three major components to it. One is a psychiatric piece, so it's often characterized by depression, personality changes. We often hear from families that, you know, dad just started acting different. He just wasn't the same person. Another component of it is what's called cognitive dysfunction. So this is the ability to make decisions, to perform daily tasks, to go to work. So you gradually lose the capacity to be able to do any of those things. And this is a truly progressive disease. So things start out very slowly. It lasts for 15 to 20 years until a person passes away from usually complications of the disease.
And if you get the disease, what are the chances of surviving it?
There is no chance of surviving. It always ends in death. If you live long enough, you will get symptoms of the disease. It's one of these truly what's called complete penetrance diseases, where that gene, that mutation will show up.
You've interacted a lot with the HD families, and the kind of toll that takes of seeing these people suffer.
When you watch people you care about getting sicker by the day, and you know, what stands between them not getting sick is the research that we do. It's unbelievably motivating.
Frances Saldana, our friend and colleague, talked about her daughter Marie's death.
Yeah.
It sounds horrific.
It was
Grand mal
hideous.
seizures.
It was horrible.
Yeah.
It was just horrible. And she, you know, I knew Marie when she was still walking around and talking and enthusiastic about life, and then, you know, watched her progress till she was in this hospice situation and just in extreme pain. And I think that was something that really hit me. I didn't fully appreciate some of the pain, the physical pain that was involved in it. They can't swallow, they can't speak, there's a lot of eye movements, and this happens all day long except when they're asleep at night.
1998, Jamie Thompson, actually at the University of Wisconsin, describes for the first time human embryonic stem cells. What did that science represent for a disease like HD? It
provided the opportunity for the first time that you might be able to study the disease in a human cell. It also provided the hope that you might be able to use these cells for transplantation.
You have a picture of a normal brain and a brain of a HD patient.
Yeah.
I have described the brain of an HD patient as looking like a piece of Swiss cheese.
Yeah.
So the hope is that transplanted stem cells could slow or halt that process.
Exactly. That would be the goal, to maintain the intactness of the brain.
For the patients and their families, the clock is ticking. If someone has the mutant HD gene, it's really only a matter of time.
Absolutely.
So there is an urgency. How do you jump on a scientific discovery like that, an opportunity, rapidly?
The ability to collaborate with other investigators that have the knowledge to be able to apply it very quickly was invaluable. I mean, this stem cell center here, this community we have here, you know, that made all the difference. I couldn't have done it by myself, but the other piece you needed was funding. We've done some pilot studies to transplant neural stem cells, so cells that are going to become brain cells, into the brain of Huntington's disease mouse models. And that was 100% funded by private donations. We'll get a check for $100 or $200, and every single bit of that helps.
So far, the results of the studies in your lab suggest that stem cells transplanted into a model of HD really could affect the progress of the disease.
I think one of the most striking initial results was there's a type of symptom in the mouse where if you hold them by the tail, they just sort of hang there, an unaffected mouse. But if a mouse that has HD-type symptoms, they reach back and clasp their hind legs, and it's very striking. It's either they do it or they don't. And in this case, at this first set of time points, none of the mice did that. And I've never seen that before.
So that really exciting result then sets you up to move forward to see if this strategy would work in humans.
I'm the most excited I've been in years.
Leslie, it's been a great pleasure talking to you today. Thank you for all you do for the HD families and for the Stem Cell Research Centre.
Thanks, Peter. It's really been great.